that are in excess of the clinical need of the person seeking the fertility treatment and would otherwise be discarded unless they are used for research, or embryos which are not suitable for implantation and would otherwise be discarded can be used for research. So these are typically going to be fresh embryos, not frozen, because you do the test, you discover that they have a defective gene. We then have to figure out a way to get them from the testing lab to our research labs. And that's possible. There are ways to transport the embryos. They're just destroyed now because if you have an embryo that has Tay-Sachs disease and it's not suitable for implantation, it simply gets destroyed. But if we wanted to study Tay-Sachs disease, a wonderful way to study it is with this embryo. So Gary Smith in the Department of Obstetrics and Gynecology is looking at ways to facilitate transporting these embryos for purposes of research. So we're going to be looking at two kinds of embryos, and it's important to understand this. One kind of embryo is the embryo that is not suitable for implantation, has a defective gene. The others are perfectly suitable, but it is not needed by the person seeking the fertility treatment. It is excess. So it's those two kinds of embryos. The other thing I forgot to do was my show and tell. I haven't done this since first grade, so I forgot how you do it. This is today's Michigan Daily, and the headline is, Professor's Stem Cell Trial Gets FDA's Okay. This is landmark research going on at the university, and Jack will tell you about it, and I'm ceding the podium.
I had a question back here.
Oh, I'm sorry, sure.
Yes. The in vitro fertilization process, that sounds like it's rather wasteful since you're the bioethicist. Isn't there a process they can look at improving it where there's not so many wasted embryos in the process?
No, it's not a question of what. The question is, the question is the in vitro fertilization process wasteful because it produces more embryos than the couple needs? And the answer to that is it depends. In some cases, all the embryos that are produced are necessary to produce a successful pregnancy. There's only a 33% success rate. They might have to come back for a second time. What we don't want to do is have to create new embryos every time, because that means harvesting eggs from the woman, that means creating the embryos all over again, that means hormonal stimulation of the woman. That, on balance, life is a balance, on balance, it is better to produce several embryos the first time around so that you have them available should you need them than it is to subject the woman to repetitive hormonal changes and production of the eggs. Again, I am getting into the scientific area, and Jack will correct me, but that's about, yeah. And no, it's a legitimate question, I understand, but the only way you can be sure, the purpose of in vitro fertilization is to work in the area of reproductive medicine and produce children for couples who are otherwise childless. And so you want to maximize that result.
Still sounds like a wasteful process.
Well, I understand.
Thanks, Ed. Ed stole a lot of my thunder, but I'll still continue and give you with what information I have. And it's no accident that the science really follows kind of the guidelines and policy, much like our presentation today, because everything we do in the lab, specifically as it pertains to embryonic stem cell research, is under the direction and guidance of policies and guidelines established now both federally, nationally, and institutionally. And so before I kind of get into the trees, I'd like to give you the overview of the forest of, well, why are we here to begin with? Why is it that stem cells are important and that we're continuing to learn about them and experiment with them? And there are really 4 reasons why stem cells are important. The first is that they can enhance our understanding of human development. Obviously, embryos come from a very early stage in development, which give us the potential to study that process in normal development, but also embryos that might have specific mutations like Huntington's disease, which Ed referenced earlier. Secondly, as we understand the origin of development, that also gives us insight into the origin of disease. Disease is really, a lot of inherited diseases are often development broken down or gone awry. So as we understand how things develop normally and consequently abnormally in certain diseases, we then gain insight into how we can fix those problems. A more well-known, probably the most widely known aspect of stem cell research is that of cellular therapy, where dead or dying or defective cells or tissues in the body are replaced by cells that have been created in a petri dish, transplanted into the body, then taking over the function of those cells that are dead or dying, such as in diseases like Parkinson's or diabetes, for example. And the fourth aspect of stem cell research that's, which they provide some use, is for drug therapy or drug development, drug discovery. We can take stem cells and specialize them into heart cells or brain cells, whether they're normal or have mutations. And then in a dish, in a laboratory, a pharmaceutical company could take and screen, hundreds of thousands of developmental compounds to see whether or not they're toxic to these human cells, or whether or not they might replace or correct the defect that they have in the beating in the case of heart cells. There are a number of uses and reasons why we're studying stem cells. And so the first question is, well, what are stem cells? And a one-sentence definition that encompasses all stem cells is that they're unspecialized cells that have the potential to give rise to specialized cell types, like the kind I've shown here, such as blood cells or muscle cells or brain cells. And that applies to any type of stem cell. And all stem cells share two important properties, and they really need to meet these criteria in order to get the definition of stem cell. And one is that they can generate more stem cells. And so here's a schematic of a cell up here. And I brought slides. I know Ed didn't bring slides, but I figure it's probably been a while since most people in this room have had the biology back in college and high school. So hopefully this makes it a little more meaningful and memorable. So I've kind of drawn a cartoon of a stem cell up here. And one of the properties a cell has to demonstrate to be a stem cell is the ability to regenerate itself, or what we would call self-renewal. And so that's demonstrated here, where it just divides and gives a copy of itself. But the other important property, which it has to meet in order to be characterized or defined as a stem cell, is the ability to give rise to those specialized cell types. So in this case, this cell here, under the right conditions, may give rise to blood cells, muscle cells, or neural cells. All stem cells demonstrate that ability. But the extent to which they demonstrate that ability depends on the type of stem cell you're talking about. And so for today's purposes, I'm going to talk briefly about three types of stem cells, just hit the highlights to give you an idea of what types there are. And the first are adult stem cells, and I'll talk about these in more detail in the subsequent slides. Embryonic stem cells, and a new type of stem cell called induced pluripotent stem cell, or IPS. So I want to first talk about the adult stem cells. And adult stem cells is kind of an umbrella term, which captures all non-embryonic stem cells. So that would include stem cells that come from the cord blood, the placenta, the infant, the senior citizen, whomever. Any non-embryonic stem cell is categorized as an adult stem cell. And they're present in many tissues. And during development, they actually form those tissues. And during the adult or the lifespan of the organism, these stem cells in some tissues provide maintenance and repair, or homeostasis, to make sure the system functions normally over the lifetime of the organism. And that's especially true for the stem cells in the blood that reside in the bone marrow. They exist for the lifetime of you and I and the animals we study. But stem cells are not found in all tissues, adult stem cells, such as the pancreas here, which is involved in diabetes. That organ doesn't have any adult stem cells as we know of today. Adult stem cells do maintain the ability to generate specialized cell types, but they only do it in the tissue in which they come from. So for the stem cells that we know exist in the brain, those stem cells only have the ability